Greetings everyone. So today we are going to discuss about the growth in bacteria. So growth in bacteria here refers to the collective growth of the population of the bacteria rather than the size of the bacteria. So here we are going to discuss about the growth of bacteria in different phases, such that here growth of bacteria occurs in six different phases. Generally, we can conclude those six phases into four phases, but according to the recent research, uh, some microbiologists have classified the phases into six phases. We're going to discuss them in this condition. And here, growth here terminology not refers to the size of the bacteria, increase in the size of the bacteria, but rather the increase in the population of the bacteria, increase in the population of the bacteria within given interval of time. So when it comes to the first phase of growth, it includes lag phase. Lag phase here, the word lag refers to delay or slowness, such that this is a phase in which there is no growth of bacteria, there is no cell division occurring, growth rate is zero. There will be only the increase in the size of the bacteria, and will be the process in which the bacteria will build up their protoplasm, which means there will be increase in the volume and mass of bacteria. Volume and mass of the bacterial cells will be increased, and thus their protoplasm will be built. After that, uh, there are certain conditions such that bacteria will develop their metabolic activities, which means the rate of metabolic activities would be maximum in lag phase, such that metabolic activities and all of the preparatory phase would be considered within the lag phase, such that all of the metabolic activities, including all of the synthetic processes, will be maximum in the lag phase rather than other phases, because the cell will be preparing itself in order to divide. So uh, that is also the phase in which there will be the preparation of enzymes. There will be preparation of enzymes for cell division, as well as, sorry, enzymes, as well as the intermediate word refers to the intermediate phase of cell division, S phase of cell division, during which there will be the preparation of the cell division would also occur in the lag phase. And in the lag phase, all of the metabolic activity will have the highest rate. Metabolic activities will occur, but there will be no increase in the population, no cell division will occur at all. And population just remains constant, population remains same, no change in population. Moreover, in this case, there will be neither the rate of division nor there will be the rate of death of bacteria. After that, when it comes to the second phase, which we are having, second phase would be the exponential phase or log phase. And in this case, exponential phase can be explained in such a manner that if there is, let's suppose, a particular bacterial cell, and that bacterial cell divides into two bacterial cells within 20 minutes. Let's suppose it divides, one bacterial cell divides into two bacterial cells in 20 minutes. Now these two bacterial cells divide into four bacterial cells. One, two, three, four bacterial cells. As a result of which, this also requires around 20 minutes. After that, four bacterial cells divide into eight bacterial cells, and that also requires 20 minutes. So in this process, when the bacterial cells are being doubled, the population of the bacterial cells is being doubled within a regular interval of time, then at that moment, this type of growth would be considered as the geometric growth or exponential growth. Apart from which, if there would be a condition in which if a bacterial cell divides into two bacterial cells, and then instead of the two bacterial cells dividing into four bacterial cells, one of the bacterial cells uh, becomes unable to divide, is remains unable to divide due to some unfavorable circumstances, due to any unfavorable conditions or environmental factors, such as depletion of nutrients, exhaustion of nutrients, or for presence of any toxic substance within the environment. Three of the bacterial cells are formed within the third turn or the second turn, as a result of which, when this type of progress or this type of growth, in which the growth rate is not being doubled but being increased in an arithmetic way, such that previously there was a growth of one cell, after which there is growth of another cell. I mean, previously there was one cell which was grown, after which there was one more cell grown, after which there would be another cell grown, due to which there would be around an arithmetic sequence occurring, due to which we'll call that as arithmetic growth, right? Arithmetic growth would be there. After that, when it comes to the exponential phase or log phase is the actual phase in which the cells, being which are completely prepared for the cell division, will start dividing themselves at a rapid rate, such that the population will double with the doubling time, or double with the generation time, such that the doubling time, which is the time required for the bacterial population to double itself, is known as the doubling time or generation time. Now, within that time period, if the bacterial population is being doubled, then at that moment, this would be considered as an exponential growth, and the flow or the phase would be considered as an exponential phase. Now, exponential phase basically has the growth rate as a constant. Growth rate is constant. But in between the lag phase and exponential phase, we are also having another phase which is known as lag phase and uh, lag phase and exponential phase. There is a phase which shows acceleration of in the growth of bacteria, and that phase which has an acceleration of the growth of bacteria is known as the acceleration phase, and that acceleration phase showcases an increase in the growth rate, after which this linear relationship which is shown between the log of the number of bacteria and the time period in which the bacterial cells are being divided is known as the exponential phase, in which the growth rate is constant and is increasing constantly with regular interval of time, as a result of which there's a linear relationship formed between the log of number of bacteria and the time period, right? And here the rate of growth is maximum. Rate of growth is maximum. One more thing, the metabolic activities, such as metabolic growth, is start started from the lag phase and then goes on till the death phase, until the cell divides. After that, we are having another phase over here which is known as retarding phase. Retarding phase is a sort of phase in which the growth rate is decreased due to a number of reasons in the exponential phase, such that if there is one of the two conditions in the exponential phase, which first involves form presence of any toxic substances within the environment which inhibits the cell growth, or the presence or depletion of any sort of nutrient in this environment, or depletion of nutrients in the medium, which will cause the death of the bacterial cell ultimately. So either there would be a depletion of the nutrients, or there would be the presence of any toxic substances within the environment which inhibits the cell growth. Then at that moment, the growth rate, cells become unable to divide, right? After that, grow cells become unable to divide, apart from which cells become unable to divide. Apart from which, cells become unable to divide. After which, the cells become unable to divide, or in this condition there can also be lack of oxygen for oxygenic bacteria. If there is lack of oxygen, then these aerobic bacteria are unable to survive within this condition, which are some unfavorable conditions which prevent the growth of the bacteria, or makes them unable to divide. After that, in that case, if these conditions persist in the exponential phase, there would be a phase would be a retarding phase in which the growth of the bacterial cells will start to decrease. Growth rate would be starting to will start to decrease, such that growth rate starts to decrease, and there will be uh, death of the bacterial cells occurring, as a result of which there would be a certain condition in which the of rate of division in the retarding phase, the rate of death in the retarding phase, the rate of division will be greater than the rate of death, while gradually, when time will pass on and any of the unfavorable circumstances within the medium will persist, it will cause the cells, it will cause the bacterial cells to start dying rapidly, as a result of which there will be a condition in which the rate of division will be equal to rate of death, due to which there would be a phase in which the growth rate will again become zero, due to equal rate of division and rate of death, due to which the phase obtained in this condition would be the stationary phase, which would be a stationary phase, or in other words we can also call it as a maximum stationary phase. After the maximum stationary phase, gradually the death rate will start to increase, and the bacterial cells start will stop multiplying, as a result, due to which the death rate will now become greater than the rate of division, as a result of which death rate being greater than the rate of division will cause the rate of growth to become negative, due to increase in the rate of death and decrease in the rate of division. There would be a negative growth rate, which would be considered in the death phase or decline phase, in which the population will be declined or dead, due to which there would be a negative growth rate would be there, right? Negative growth rate. Now, within the death phase, some of the bacteria will also tend to form endospores, exospores, or cysts, in order to uh, persist their life cycle within that environment. But again, those metabolically inactive or dormant bodies will not be able to divide themselves and increase their population. So that would be, even though considered as living, but will remain static or constant in growth rate, which means that won't be able to divide themselves, as a result of which they won't be able to grow themselves, as a result of which the growth rate here would be considered as negative, as there is no possibility of dividing endospores, exospores, or cysts in order to form the bacterial cells. For that, they require favorable conditions, as a result of which, um, even though they would be considered as living, but again, they won't be able to divide, and growth rate would be again considered as negative. So this is all about today's video. Hopefully, we'll meet again in another video.